All right, let's talk about A-level biology practicals. I know, the pressure can feel pretty immense. But honestly, these biochemical tests, they're where you can pick up some really solid, satisfying marks, but only if you've got the method down cold. So that's what we're going to do. Think of this as a quick, focused revision session to get you feeling super confident for exam day. So here's the plan. We'll kick things off with the basics, then jump right into the sugar tests. After that, we'll nail the tests for starch and protein. Then we'll level up and look at how to get proper quantitative data with a calibration curve. And finally, we'll cover the common mistakes everyone makes and, of course, how to stay safe in the lab. Ready? Let's get into it. You know, this is really the million-dollar question, isn't it? It's one thing to see a test tube change color, but it's another thing entirely to know exactly what that means, how to describe it perfectly for the examiner, and how you got there without tripping up. That's the skill we're going to lock in today. Okay, first up, the big one, the Benedict's test. This is probably one of the most fundamental tests you're going to face, and it's your absolute go-to for identifying reducing sugars. We're talking about all the monosaccharides, like glucose, and some disaccharides, like maltose. It all works because they have a special chemical group that's free to donate electrons and reduce the copper ions floating around in the Benedict solution. Now, the method itself seems simple, but getting it right is all about precision. You take your sample, you add exactly the same volume of that blue Benedict solution, and you pop it in a hot water bath. And don't rush the heating. That's the magic step. It provides the energy for the whole reaction to happen, for the sugar to reduce those copper ions. That's what gives us our color change. And here's the really cool part about interpreting the results. This test isn't just a simple yes or no. Nope, it's actually semi-quantitative. The color it turns gives you a pretty good estimate of the concentration of sugar. If it stays blue, there's nothing there. But if it goes green, yellow, orange, all the way to a thick brick red, well, you know you've got a lot of reducing sugar. You've got to memorize this color scale. Ah, but what happens when it just stays blue? You might think, okay, no sugar, but hold on. It just means no reducing sugars are present. You could easily have a non-reducing sugar in there, like sucrose, where all the reactive bits are tied up in a bond. So, how do we find out if that's the case? Okay, so this is a bit more involved. It's a two-stage process. First, you've got to break that non-reducing sugar down into its monosaccharide building blocks. We call that hydrolysis. You do it by boiling a fresh sample with some dilute hydrochloric acid. That's what snaps the bonds. But, and this is the most important part, the bit everyone forgets, Benedict's solution just doesn't work in acid. So you absolutely have to neutralize it first. Add some sodium hydrocarbonate, get it back to neutral, and then you can do the Benedict's test again. If you get that color change now, you know you started with a non-reducing sugar. All right, let's switch gears and look at our next set of essential biochemical tests. Now we're on the hunt for two other huge biological molecules, starch and protein. The good news is these are pretty straightforward qualitative tests. They give you a clear yes or no answer. The test for starch, it honestly couldn't be simpler. You just take your sample, add a couple of drops of iodine solution, which is normally a kind of orange-brown color, and give it a mix. No heating, no waiting around. The reaction is instant if there's starch in there. And the results are just unmistakable. A positive test for starch gives this really intense, deep blue-black color. It's pretty cool, actually. What happens is the iodine molecules get trapped inside the spiral shape of the starch molecule. If there's no starch, well then nothing happens, and the solution just stays that normal orangey-brown iodine color. Easy peasy. Okay, on to protein. For this, we use the Biuret test, which is looking for the peptide bonds that link all the amino acids together. Now, just like with the non-reducing sugar test, the conditions here really matter. You have to make the solution alkaline first, so add an equal amount of sodium hydroxide, then you can add your copper sulfate. The results here are also super clear. If you've got protein, the solution turns from its original blue to this really distinct lilac or purple color. If it stays blue, that means no peptide bonds, so no protein. Quick tip, if you hold your test tube against a white tile or a piece of paper, it makes it much easier to spot that subtle color change. So, we've covered qualitative and semi-quantitative tests. But you know A-level biology, it's always pushing for more precision. So let's get into a really key required practical, turning our Benedict's test into a fully quantitative tool so we can find the exact concentration of something like glucose in a fake urine sample. And the tool we use for this is the calibration curve. What is it? Well, it's basically a reference graph. You create a bunch of samples where you already know the exact concentration, you test them, and you plot the results. This graph then becomes your key to unlocking the concentration of any unknown sample you have. So, how do we actually make one? First, you need to create a dilution series. That's a set of glucose solutions with different known concentrations. Then, you run the Benedict's test on every single one of them. But here's the game changer. Instead of just looking at the color, you use a colorimeter to measure how much light is absorbed by the solution. The more sugar, the more precipitate, the more light gets absorbed. You plot those absorbance numbers against your known concentrations, and then you draw a line of best fit. That's your curve. And this right here, this is why this method is so important. This is a classic exam question, so listen up. Using a colorimeter is what takes your experiment from just kinda sorta to bang on accurate. It gives you a number, a proper absorbance reading. It completely gets rid of you having to guess, is that more orange or is it brick red? No more of that subjective stuff. It makes your results precise, reliable, and properly quantitative. That's where the top marks are. So once you've done all the hard work and plotted your calibration curve, using it is actually the easy part. You take your unknown sample, you run the exact same Benedict's test, and you measure its absorbance with the colorimeter. Then, you just find that absorbance number on the y-axis of your graph, go across to your line of best fit, and then drop straight down to the x-axis. And boom, there it is, the precise glucose concentration of your unknown. Okay, we've gone through all the methods. Now let's wrap this up by making sure you don't drop any easy marks. We're going to look at the most common mistakes and, more importantly, how to do all of this stuff safely. These are the absolute classic mistakes you need to avoid. Number 1, just describing a color when you could be using that semi-quantitative scale. Number 2, and this is the really big one, forgetting to neutralize the acid in the non-reducing sugar test. Your test just won't work. Number 3, being sloppy when you make your dilution series. If those known concentrations are wrong, your whole calibration curve is useless. And finally, just a simple slip-up like misreading the scale on your graph that can undo all your amazing work. Be careful at every single step. And of course, safety is just non-negotiable. Benedict's solution can be an irritant, so eye protection is a must always. You're going to be using hot water baths, so use tongs, be sensible, and avoid scalds. And it sounds obvious, but keep your glassware away from the edge of the desk so you don't have to deal with breakages and cuts. And this is the golden rule of any lab. I don't care how small it seems. A tiny spill, a cracked test tube, your first move every single time is to let your teacher or the lab tech know what's happened. Always. And there we have it. We've walked through all the key tests, from those simple color changes right up to getting precise quantitative data. You've got the methods, you know what the results are telling you, and you know the common traps to sidestep. You're all set for these practicals. So I'm going to leave you with one last question to kind of tie all this together. Think about an enzyme, let's say amylase. We know it breaks down starch into maltose. Now, based on everything we've just gone through, how could you use these tests to actually measure the rate of that reaction? See how fast it's happening. That's when this stuff stops being just a lab test and starts becoming a really powerful tool for understanding biology. Give that some thought. Good luck.